This is part 4 of the chapter 13 lecture notes, and we should finish up in this section. Okay, so we are now on section 3, and we're talking about DNA mutations. So mutations that occur during replication are called spontaneous mutations. They're spontaneous. That means they just happen spontaneously. There's no reason for them. There's no chemical or dangerous something in the environment, like radiation, nothing like that causes it. It just happens during replication. When DNA is replicating, there is a mispairing of bases. Another thing that can happen is transposons, which are, we used to call them jumping genes. They can move from one chromosome to another, or from one location on a chromosome to another. But these are just pieces of DNA that can actually move. So mispairing during replication or transposons, these are the causes of spontaneous mutations. So DNA polymerase not only adds nucleotides to the old strand, forming a new strand, as it does that, it proofreads the new strand. And it will find errors. So say there's one error in every 100,000 bases, it will find those errors, and generally it will correct those errors using DNA, repair enzymes, DNA repair enzymes, I think is what they're called. But anyway, because of that, instead of there being a mutation in in one of every 100,000, the overall mutation rate is actually 1 in every billion nucleotide base pairs. And that is because of the fact that DNA polymerase can proofread and correct errors. So spontaneous mutations occur without cause, basically. They happen during replication or because of transposons. Induced mutations occur, they're caused by mutagens, which can be radiation or chemicals. If they lead to cancer, they are called carcinogens. Carcinogens are cancer-causing mutagens. There are environmental mutagens like food and tobacco smoke. Tobacco smoke is responsible for 1/3 of all the cancers. There is a little section on how a chemical is tested for mutagenicity, and it's a test called the Ames test. I didn't ask any questions. I don't think there's not any questions on the study guide, but the Ames test exposes bacteria to the mutagen. That would be your experimental group, and then there's a group of bacteria that are not exposed to the mutagen. That's the control group. So if there is bacterial growth, then the mutation did occur. And the mutation did not occur in the control group here, and it did occur in the experimental group. But basically, I know it's kind of hard to see from looking at this illustration, but one group is exposed to the chemical mutagen or the suspected chemical mutagen, and then that's called the experimental group. And then the control group is not exposed. And they're both plated on petri dishes and incubated, and if the bacteria does grow, then the Ames test says that it's positive for mutagenicity. So anyway, effective mutations on protein activity. So, okay, because any mutation can lead to a change in the amino acid sequence of the protein. A point mutation is the least, I guess, dangerous or detrimental type of mutation. One type of point mutation is called a base substitution. So if the base is a T, maybe it's substituted with an A. It's a change in a single nucleotide base. So it changes one codon or triplet code on the messenger RNA to a different codon. So maybe it's AUG, start codon, changed to AUC, which would not be a start codon. But effects on the protein are going to vary. Sometimes it makes the protein non-functional. In the case of cystic fibrosis, that's an example where one base substitution actually leads to the hemoglobin molecule being non-functional, and the red blood cells are sickle-shaped instead of round, and that leads to all of the symptoms that we see, the terrible symptoms that we see in sickle cell anemia. But then we can have just reduced functionality. So the protein's okay, but something's wrong with it, but you wouldn't call it non-functional. Then we have what's called silent mutations. This is a mutation in the DNA sequence that actually does not affect the protein at all. But frame shift mutations would be when you have an addition or a deletion within the DNA sequence. So either a nucleotide is inserted or it's deleted, and it will shift the reading frame from that point onward. So the protein in a frame shift mutation is always rendered non-functional. They have used an analogy here that can help you see just how bad the change is. They didn't give you an analogy with a point mutation, so let's just let's just say I'm going to give you an analogy with this normal sentence, the cat ate the rat, okay? So if the cat ate the rat were to undergo a point mutation, let's just say the R in rat is changed to a B. That would then say the cat ate the bat. That doesn't change the sentence that much. A bat and a rat are actually kind of related to each other. They're not the same thing, though. So that would be like a protein having reduced functionality. But if you have a frame shift mutation, let's say that the C is removed in the normal, the C is removed. So now we have ATA, the ATA, tet, her, at. That's not even the same sentence. I mean, that there's no comparison with that. It's not even a sentence at all, And then if you insert an extra C here, then you can see where cat ate the rat is, but it's now shifted. So it's the ka, tat, eth, erat. That's not a sentence either. Those are not even words. So after a deletion or an insertion, from the point where the deletion or the insertion occurred, you have a non-functional, you have a whole different sequence of amino acids. So here's an example for a point mutation. This is the only point mutation here. These are frame shift. The addition and the deletion are frame shift mutations. So the title of the slide is misleading. But here's a point mutation. So what we see is in the codon UUU in the normal messenger RNA, we see a point mutation where the first U is a C. And UUU codes for phenylalanine, whereas CUU codes for leucine. So in this case, the point mutation called a base substitution did change one amino acid. Maybe that's a bad thing. Maybe it doesn't even matter. Maybe the protein, isn't really changed all that much. But it could even lead to a non-functional protein. But it did change just one amino acid. And in a protein, you can have hundreds of thousands of amino acids. So let's look at an addition. So here we have an addition of an A. Up here in the normal sequence, it was ACG. Now we have in front of the ACG, we have an additional A. So that changes the entire reading frame, and it makes now the amino acids asparagine, I think that's glutamate and another asparagine, whereas up here there were threonine, arginine, and isoleucine. Totally different amino acids. The deletion that occurred, it was a UUU, and one of the U's is deleted off the end. So now it becomes UUA, and that changes the second, third, fourth, and fifth. I didn't mean to highlight methionine. I don't want to go back to that though. So the methionine didn't change, but the ones that did change were from this point on, the leucine, arginine, and glutamate, the ones that are in red, those amino acids are changed, and so basically the entire protein from the start codon on, forward is going to be totally different and probably non-functional. Okay, this is showing you the example of how 1 point mutation in normal hemoglobin, if that one, this was CTT, which is GAA, the codon is actually GAA and codes for glutamate in the normal one. But if there is a change and in the DNA it's CAT instead of CTT, then the codon is now GUA instead of GAA, and that codes for valine. And that just that one valine amino acid difference causes those red blood cells to be sickle-shaped instead of rounded, and leads to blood clotting and anemia, low oxygen levels, and just terrible pain, and the disease that we call sickle cell anemia. All right, non-functional proteins. There's a little information here about non-functional proteins. It tells about the disorder PKU, or phenylketonuria, in which phenylalanine builds up in the system and it causes an intellectual disability. Although the phenylalanine, if you remove that from the person's diet, then they can, that is a good treatment, and they can be pretty normal as long as you remove phenylalanine from, they can't eat any foods that contain phenylalanine. Another example is androgen insensitivity. That is when cells are unable to respond to testosterone. So a person who is chromosomally male, who has the XY sex chromosomes, so their chromosomes say that they are male, they will actually develop into a female because of the fact that their cells are insensitive to testosterone. It's called androgen insensitivity, and that is caused by a faulty enzyme. Enzymes, are proteins. So a faulty enzyme can cause PKU, and it can cause androgen insensitivity. Let's see, mutations can cause cancer if proto-oncogenes are mutated. They mutate and become oncogenes, which are cancer genes. When tumor suppressor genes are mutated, they don't suppress the growth of a tumor now. So that can lead to cancer. So mutations in the oncogene and tumor suppressor genes can cause the cell cycle to occur uncontrollably at an uncontrollable rate, kind of goes out of control, and then a tumor can form. And that is the end of this chapter.